guys, in today's lecture, we're going to be talking about the use of biomaterials in tissue engineering applications. So tissue engineering was actually first introduced in the 1980s. In fact, the first tissue engineering professional meeting was in 1988. And the strategies that are utilized, or at least that were utilized at the beginning in tissue engineering, came from the knowledge that had been gained in the 70s and 80s in the development of skin substitutes. From the 1990s on, the tissue engineering field matured into a more interdisciplinary field. And of course, it continues to date with many developments in trying to obviously develop better strategies to make this field a success in medicine. Some reading material that I am suggesting that you take a look at include the Biomaterial Science book, which has a whole section on tissue engineering. In addition, there's a number of review papers that are listed here. One of those that I wanted to point out was this paper by Atala, Casper, and Dr. Mikos that discusses some of the challenges of actually translating these technologies into commercial success. And then I also wanted to point out that at the bottom here, I included a website for the Air Force Institute of Regenerative Medicine. The Air Forces basically are always looking for collaborations to enable rapid and safe translation of tissue engineering technologies to the clinic for obvious reasons. All right, so what is the need for tissue engineering? So for a patient, whenever a tissue has failed, the options that are provided are to utilize extracorporeal treatments. Those extracorporeal treatments could be either of passive or more kind of active form. In terms of passive form, here we're talking about basically utilizing an extracorporeal device such as a hemodialyzer, where the blood will be circulated through a polymeric membrane that enables the exchange of molecules that need to be removed, for example, and those devices are considered passive. But as of late, there's more research into the development of systems that are not just passive, but that might also include entrapped cells, which can basically enable a function to be done. Those cells can be of human or animal sources, and those type of systems that incorporate cells are typically called bioartificial or biohybrid organs. And of course, they have the potential to be able to work a lot better than a passive system that is just based on molecular weight, for example. In addition to extracorporeal devices, we can also replace the tissue that has failed with a synthetic substitute, such as a drug delivery system, maybe like a pump for the delivery of insulin, for example, or simply by having the patient take a drug on a daily basis for the rest of their lives, or utilizing an intracorporeal device. The other alternative would be to replace the failed tissue with a natural substitute by undergoing a transplant. Of course, none of those are the best case scenario necessarily. They all have potential issues. And so this is where tissue engineering comes into place, where perhaps we are able to repair or regenerate the tissue in question to be able to provide full function to the patient without many of the drawbacks of the previous replacement options. So the goal of tissue engineering is to regenerate tissues that can be used as alternatives to donor material to repair or replace damaged tissue. And tissue engineering aims to restore tissues and organ function by employing biological and engineering strategies. Now, in order for these tissue engineering systems to be able to grow or regenerate the tissue, what they need to do is to provide the appropriate microenvironment to enable cells to be viable for cells to proliferate and cells to differentiate into the right type of tissue that has the right phenotype, the right expression of molecular markers, the right regeneration of extracellular matrix. And to do that, we have to provide those cells with spatial cues, molecular cues, and mass transport capabilities to basically simulate what a normal tissue would do. All right, the strategies to be able to develop a tissue engineering system would be A, to do a transplantation of tissue that is grown in vitro, or B, to actually grow or regenerate the tissue in situ. A is shown here on the left-hand side, and basically what we have is that we're going to be developing a scaffold, a biomaterial scaffold that could be made out of synthetic polymers or natural polymers or a hybrid type material, a nanocomposite or something like that. And that material would be placed in vitro together with cells, cell media, and factors, growth factors necessary to enable the cells to grow. All of this would be done extracorporeally in an incubator. After the tissue scaffold and the cells have incubated for a sufficient amount of time to enable the cells to grow and continue proliferating, then this complex material that now also includes cells will be transplanted into the patient. So most of the development basically is done in vitro, and then the system is transplanted into the patient with the idea that this will continue to regenerate into a fully healed tissue. In strategy B, we still develop a biomaterial scaffold, but that biomaterial scaffold is implanted into the patient without any cells. And so it just basically is the synthetic material. And the idea is that that synthetic material would include some kind of factors to be able to encourage cell migration into the tissue. So here what we're showing is cells from the normal surrounding tissue. And this is the material which is being displayed as a hydrogel or a polymer network. And what we see is that cells are being recruited to invade or to take location within the scaffold, within the material, to be able to then heal or regenerate the tissue. Those are the ideas. Of course, both of them have benefits and drawbacks, and we'll talk about some of those later on. The main components of tissue engineering constructs are going to include cells, a scaffold, and growth differentiation factors. The scaffold is going to be the biomaterial that we as engineers are going to be developing, but of course that material needs to be able to interface with cells. And in addition to that, for the cells to be able to grow, we actually have to provide growth or differentiation factors that are bioactive molecules that are going to stimulate or encourage those cells to actually function. Here we're going to start by focusing on the cells. These are the cells that we can actually include within the scaffold, and later we're going to be talking about what the scaffold properties need to be, and finally we'll talk about the way to be able to incorporate this kind of growth or differentiation factors. In terms of the cells that we can include, if we are going to incorporate those cells into the scaffold in vitro, what we can utilize is going to be either stem cells or differentiated mature cells. Stem cells are cells that have the capability of self-renewal, and they have the ability to be differentiated into multiple lineages or multiple different types of cells to be able to function as different tissues, basically. Of course, the cells of a muscle are going to be different than neural cells or that cells that are in endothelial cells, for example. And so, of course, they have to have different properties, and the stem cells would be able to differentiate into those types to be able to carry out those functions. In terms of stem cells, we have two different types. Those are going to be embryonic stem cells and then adult stem cells. Embryonic stem cells can self-renew without differentiation. They also can be expanded in culture, and they can differentiate into any cell type. So that's what makes them the most useful. Of course, there's a lot of ethical concerns with their use, ethical, religious, political, controversy with their use. But they do have benefits that cannot be overcome with other stem cells. Now we have adult stem cells, which can be of different types. There are mesenchymal stem cells, hematopoietic stem cells, hepatic stem cells, neural stem cells. They can be reprogrammed into pluripotent stem cells, meaning that they have the ability to differentiate into a number of different cell types, but it's not an unlimited number of cells of cell types. They actually have limitations in terms of the differentiation potential. One way to say it is that they are already more committed toward a specific type of cell type than an embryonic stem cell would be. These adult stem cells, the benefits of them would be that they could be obtained from the patient's own cells, potentially, and so that would be beneficial in terms of preventing rejection from utilized injection or even contamination from using donor cells from a different patient. They are much rare in terms of number. They are much more challenging to isolate and much more challenging to expand. And as I mentioned already, they're a little bit more difficult to differentiate into a specific type of cell. So they do have significant drawbacks. Among the various types of adult stem cells, mesenchymal cells are utilized quite a lot in tissue engineering. These mesenchymal stem cells are pluripotent stromal cells that can differentiate into osteoblasts, chondrocytes, myocytes, and adipocytes, among other cell lines. The sources of these mesenchymal stem cells are typically the bone marrow, the cord cells, adipose tissue, molar cells, amniotic fluid, or peripheral blood. Now, in terms of the differentiated adult cells, as expected, they're not going to have the ability to differentiate into any other cell type. Whatever they have already committed to be, that's what they're going to be. However, we can utilize things like chondrocytes, for example, for cartilage repair. One of the other problems with differentiated mature cells is that they have limited proliferation capability. They cannot be expanded in vitro. They typically begin to lose their phenotype or their properties as you start proliferating them. And the differentiation in culture is very, very difficult. However, their phenotype can be controlled either by genetic alteration or by manipulating some of the culture conditions. And so there's a lot of research into seeing, could we utilize this type of cells to actually regenerate tissues? That would be significant because they're a lot easier to obtain, basically. All right, continuing talking about the cells. So in terms of stem cells, they are known as progeny of cells that have the potential to differentiate into a variety of lineages. They were first isolated in 1981, and they were first isolated from embryos in 1998. As I mentioned before, they can self-renew without differentiation. They can be culture expanded. They can differentiate into any cell type. And let's see, I forgot what else I was going to say. Oh yeah, the types of these stem cells, we already mentioned embryonic stem cells. We also mentioned adult differentiated cells. And we mentioned that some of the sources could be from umbilical cord blood. Let's see, what else did I want to mention? I think that is about it for this slide. All right. The second part, other than the cells that we need for our tissue engineering construct, is going to be the scaffold. The scaffold, as I mentioned, is the biomaterial that we as engineers are going to be designing, and that scaffold needs to have specific properties. It needs to be tissue-like in a way. It needs to be able to encourage the cells to proliferate within it to be able to develop the tissue. And what our best understanding at the moment is that tissue engineering scaffold needs to have a behavior that is very similar to that of the extracellular matrix, which is the material, the natural material in which cells live in our natural tissues. And so right now we're going to be talking about the various properties of the extracellular matrix, which are the properties that we're basically going to try to mimic when we developed a tissue engineering scaffold. So the extracellular matrix, or ECM, is the biological material that is produced by, that resides in between, and that supports the cells. It is composed of biomolecules that are arranged in distinct structures and that are unique to each type of tissue. These biological molecules include proteins, polysaccharides, water. Some of the proteins are insoluble. Some of the proteins are soluble. It has very interesting characteristics that we will discuss in a second. This extracellular matrix also contains various factors that regulate proliferation, growth, development, and function of cells and tissues. And that's the third thing that I mentioned, that our construct needed. I mentioned that our construct would need to have, let me see, what was the word that I used? Growth and differentiation factors. These are things that are actually already present in natural extracellular matrix, but that for any synthetic material that we make, we need to actually recapitulate this or be able to include this in the material. The extracellular matrix also enables coordinated interplay of spatial and temporal physiochemical cues. And that's something that nowadays in tissue engineering field, people are trying to recapitulate or to be able to mimic. Can we actually change the properties of our scaffold over time? Can we actually control what biomolecules are presented to the cells at what time and in what locations? Because all of that is what actually guides the cells to be able to function in a specific way and be able to develop or regenerate the tissue. So our tissue engineering scaffold needs to recapitulate the complex design of the extracellular matrix for optimal tissue regeneration. This is a figure that tries to show the very complicated structure of the extracellular matrix. Basically what we have here is epithelium on the very top. We have the basement membrane right here in between the epithelium and the stromal tissue. In the stromal tissue, we have our fibroblast cells that are surrounded by a network of materials, which is extracellular matrix. Then in between that and the blood vessels, we have another basement membrane that separates the stromal tissue from now the blood vessel itself. Then we have the blood vessel, and then we have more stromal tissue here. Now, extracellular matrix, there's at least two main types that I'm trying to display in this image. One of those is the basement membrane. That basement membrane is a membrane of proteins and fibers, and I should say protein fibers and glycosaminoglycans that separates the epithelium from the underlying tissue. Then we have the other extracellular matrix, which is everything that we have here in the normal connective tissue, and that's known as the interstitial extracellular matrix, which is the extracellular matrix in between cells. In both of these circles, they basically try to zoom in into the two main types of the extracellular matrix. Here we're looking at the basement membrane, which contains type 4 collagen, laminin, proteoglycans, among other molecules. And then here they're zooming in into the interstitial matrix, which is going to include adhesive glycoproteins, is going to include cross-linked collagen triple helices, proteoglycans as well. Both of those types of extracellular matrix have many things that are similar in that we're going to have a mixture of proteins. Some of those proteins are structural proteins, such as collagen, which we see in both types. We're also going to have other glycoproteins that have the function of making that tissue be more like gel-like, be able to absorb a lot of water, be able to take some to have some elasticity and to be able to take in some stress. And those are going to be things like the inclusion of these proteoglycans, which you see in both different types. There's also differences between them in terms of the architecture. And another thing that I wanted to point out while I have this figure here is just to remind you again that the cells grow by, they actually require attachment to the extracellular matrix in order to live and to function. And you can see that here. Here, for example, we have an epithelial cell that is attached to the basement membrane through integrins. Remember that integrins are proteins that are present on the cell membrane and that have the ability to bind to specific peptide sequences on the extracellular matrix. Those integrins, remember we talked about them being able to bind to cell adhesion peptides. And one of those cell adhesion peptides was the RGD peptide. And I just wanted to point that out. That same thing is being shown here for this fibroblast that is attaching to an adhesive glycoprotein through the use of integrins. All right, that's all that I wanted to mention right now. All right, so the extracellular matrix is going to have three main types of components. One of them is structural proteins that are basically going to provide most of the mechanical strength to the material. Those structural proteins include collagen and elastin. We're going to have an amorphous matrix, which is the material that is responsible for the gel-like properties of our tissues, and that is going to be composed of glycosaminoglycans or GAGs, proteoglycans, and hyaluronan. And then we're going to have adhesive molecules that are going to enable the cells to be able to attach to the tissue, and those are going to include fibronectin and laminin. Now we're going to go in through each of these materials and discuss some of their properties. All right, so to begin with, we have collagen here. Collagen is actually the most abundant structural protein in the human body. In fact, about 25% of the human's protein mass is based on collagen. There's more than 29 different types of collagens in the body, but out of those, 90% of the collagen is actually one of these 5 types, 1 through 5. Number 1 is ubiquitous in both soft tissue and hard tissue. It is found in skin, bone, tendons, ligament, internal tissues, etc. Type 2 is found in cartilage, intervertebral discs, vitreous humor of eye, and other locations. Type 3 is typically found in soft tissues, especially in healing tissues and in the walls of hollow organs, such as blood vessels. And it's typically found along with type 1 collagen. And then type 4 is found in the cell basement membrane, and type 5 is found in the hair and placenta. Out of those types, I would say that type 1 is the one that is most widely studied in terms of its use in tissue engineering scaffolds. And that's the reason for it is that the fact that it's found in both hard and soft tissues, and so we can utilize it for the development of different types of materials for different applications. Now, in terms of the properties of collagen, it has great tensile strength and great elasticity, and you will see that in the next 2 slides where we talk about the actual structure of it. Before I move on to that, I just wanted to mention 2 more things. One of it is that collagen has a structure that has been preserved across many species, which means that we could actually utilize collagen from other sources other than human with little immunogenicity when transplanted. So that's something that's really beneficial. Another thing that I wanted to mention is that collagen has significant applications. It has been utilized not only as a bioscaffold for tissue engineering applications, but it has also been used in cosmetics, blood vessel development, like artificial blood vessel development, in drug delivery systems, and in sutures. All right, so we begin by looking at collagen. So collagen, as we said, is one of the structural proteins of the extracellular matrix. One of the most characteristic things about collagen is the frequency by which this sequence of amino acids appear in the collagen structure. The sequences are glycine, proline, and X, where X represent any other amino acid, or glycine X HIP, which HIP stands for hydroxyproline. And the structures of those are shown here on the right-hand side. As you can see, both of the sequences that are important include glycine, and glycine is present in very high numbers among these collagen structures. And one of the key things about this is that this is the smallest amino acid that actually has no side group. It's actually the smallest amino acid, which enables it to be able to tuck in the twists of the triple helix that these collagen peptides are going to be forming. The collagens or the precursors to collagen are synthesized in cells as soluble precursors, but they are then secreted to the extracellular fluid where they then begin assembling. The assembly begins by taking 3 of these linear amino acid chains or linear polypeptides, and each of those are going to form an alpha chain. That alpha chain again is basically an alpha helix that it's made from the strands of the collagen molecule as it twists together to form tropocollagen molecules. So these 3 are going to twist together to form what's known as a tropocollagen molecule. That tropocollagen molecule is held together through hydrogen bonding. Then those tropocollagen molecules or macromolecules now assemble into strands that are about 300 nanometers in length and are separated by about 40 nanometer gaps. The strands are organized into a staggered array as is shown here, and they form collagen fibrils now that are about 1 micrometer in diameter but can be tens of micrometers in length. The tropocollagens are also cross-linked chemically, as you can see in this graphic, through enzymatic reactions. And so we do have a system that is basically insoluble now due to the cross-linking and due to the large structure and the large size of it. Now, one thing that I want to mention is that you can see the significant order of this protein. We have the primary structure, which is just the sequence of the amino acids, which is going to dictate how they are now going to assemble into alpha chains. From those alpha chains, we then have the secondary structure, which is going to be the local configuration of the polypeptide chains that results due to the kind of twisting of that molecule and the hydrogen bonding in between the peptide sequences. We then have the tertiary configuration, which is the formation of this tropocollagen triple helix, which it's also held together by hydrogen bonding. And then finally, we have the quaternary structure, which is basically the supramolecular structure of all of these tropocollagen helices coming together to form the microfibril. Now, in terms of the properties of this collagen, the main thing says that it is non-extensible, so it cannot be pulled into extending. It does not expand, and it is incompressible. All right. Here's a scanning electron microscopy image showing the structure, which basically is exactly like we showed before. We have the initial peptide chains that form the triple helices. Those triple helices form tropocollagen, and from there they assemble into fibrils and then even larger structures. This is here a transmission electron microscopy of collagen as well. The other protein that is of importance in terms of structural proteins is elastin. Elastin is a non-soluble protein in the human body. It is hydrophobic, it is cross-linked chemically. but it does provide elasticity. So you can think about elastin as almost like a gel that has randomly coiled peptide chains that are chemically cross-linked, and that enables it to be able to expand, to provide elasticity. As you can see, it can be stretched. In fact, it can be stretched up to 200% strain before it is completely straightened, and then it can actually be relaxed and go back to the starting configuration. It has low stiffness. The most common amino acid sequence is valine, proline, and glycine, and the chemical structures of those peptides are shown here on the right. And then X, where X is going to be any amino acid other than proline. The function of elastin is going to be to provide resiliency and extensibility to tissues. Sorry. Elastin is found in vertebrates, in the vascular tissues, in intervertebral discs, and in the skin, arterial walls, the lung, and in ligaments. Sorry. In terms of the applications of elastin, cosmetics is one of the applications that has risen in recent times. And also it can be utilized for wound healing applications because it provides that natural elasticity to tissues that need to be developed. In addition to the extracellular In addition to the structural proteins, we also have the amorphous matrix, which is basically an amorphous substance that typically consists of carbohydrate-bound proteins that has the function of providing resiliency and lubrication and resisting compression to the tissue. To begin with, we talk about the glycosaminoglycans that are part of this amorphous matrix. These glycosaminoglycans, or GAGs, are highly charged long linear alternating disaccharide copolymers that are found on cell surfaces and in the extracellular matrix. When I say charged, typically they're going to be sulfated, meaning they're going to be having these sulfate groups, which tend to be negatively charged, even though the charge is not shown here. Typically, the hydroxyl group would be off. In addition, they are linear and alternating, and I will be talking about two examples and telling you about the composition of those, but you can just look at the chemical structure and see that the first repeat unit that is shown here, or the first monomer here, is different from the next one that is shown. And so this continues alternating as the chain continues. They are found typically covalently attached to proteins as opposed to being in soluble form, although some of them are found independently. And when they are attached to proteins, they are known as proteoglycans, and they would have a structure similar to a bottle brush, where basically the glycosaminoglycan extends from the protein itself. They form viscous hydrated gels that resist compression and provides resiliency and lubrication to the tissues. They can also be enzymatically degraded. That's something that's important. The various types of glycosaminoglycans include hyaluronic acid, which is shown here on the top right. This one is the only non-sulfated protein-bound glycosaminoglycan. We also have dermatan sulfate and chondroitin sulfate. Give me a second. Heparan sulfate and heparin on the right-hand side, and then keratin sulfate. We're going to be focusing on the two that I have on the right-hand side. So beginning with hyaluronic acid, as I already mentioned, it is the only non-sulfated GAG. In terms of the structure, on the left we have glucuronic acid, and on the right we have N-acetylglucosamine. It exists in much higher molecular weight than the other GAGs, and as I mentioned already, is not typically found as a proteoglycan, meaning that it's not typically bound to proteins. It is typically found in loose connective tissues and in joint fluids, where it acts as a lubricant, and it has been utilized a lot in wound healing applications. In terms of the use of it in tissue engineering, It has been very appealing because of its biodegradability and biocompatibility and its ability to support cell growth and proliferation. So we already actually saw a paper where hyaluronic acid was one of the polymers utilized. The second one is heparin. Heparin is actually an anticoagulant. It is a negatively charged GAG made from uronic acid and N-acetylglucosamine, right here, or N-acetyl digalactosamine. Heparin was actually one of the first molecules to ever be covalently attached to a biomaterial because of the ability to act as an anticoagulant. So we can actually utilize it to functionalize the surface of materials like stents or others that that would be in contact with blood to try to prevent coagulation from happening. In addition to structural proteins and amorphous matrix, we also have the adhesive proteins or adhesive molecules that enable the cells to attach. Two of those adhesive molecules are going to be fibronectin and laminin. And just to remind you, the way that cells bind is by utilizing integrin proteins that are found on the cell membrane. So this is my cell on the bottom. The integrin molecules are then going to attach to specific proteins that are found in the extracellular matrix, namely fibronectin or laminin, which present specific peptide sequence that the integrins can recognize and bind to. And the fibronectin or the laminin are basically going to act as a linking molecule, linking the structural proteins of the extracellular matrix to your cells. So fibronectin is a glycoprotein. It presents binding sites for other extracellular matrices, matrix components as shown here. It enables the cells to bind to the extracellular matrix via self-sulfated integrins, cell surface integrins, sorry. It is a crucial component of blood clots and it is synthesized by many types, but the main form that is found circulating is made by the hepatocytes or liver cells. Laminin, on the other hand, is abundant in the basement membrane. It is tough, thin, and sheet-like, and it is where the epithelial cells are attached to. It is arranged in a form of an elongated cross with individual chains linked by disulfide bonds, and portions of it bind to type 4 collagen, to heparin sulfate, or to self-surface integrins to enable cell attachment. On the right-hand side here, I have a little table that shows some of the peptide sequences that are found in fibronectin and laminin and that are important as cell adhesion peptides enabling cells to attach. And I just wanted to remind you of the RGD peptide motif, which is the main one that has been utilized in biomaterials to develop tissue engineering scaffolds that enable cell attachment. So we can either utilize one of these proteins as part of our tissue engineering construct, or we can include some of those sequences as synthetic polypeptides to be able to enable cell attachment to take place.